you. So hello everyone, I'm Balazs, I'm a postdoc at Simulamet and thanks very much for having me. Okay, so we focus on the exploratory analysis of microbiome abundance data collected through longitudinal studies with repeated sampling of the same subjects over time. So this is perhaps a little bit more downstream than most of the talks today, but I hope it's still going to be interesting. Now, this type of data poses many challenges, including the high dimensionality, compositionality, sparsity, etc., the well-known properties of these sequencing data, but also the correlation structures induced by the repeated sampling. And recently, tensor decompositions, together with the appropriate pre-processing, have been proposed to overcome many of the challenges that this type of data poses. Okay, so before I get to the tensors, to introduce the basic concepts, consider decomposing a cross-sectional data set, subjects by taxa matrix X, into lower dimensional factor matrices U and V transpose, or equivalently to the sum of R rank 1 outer products, R being the number of components. So to uncover latent structure in a cross-sectional data set. And PCA is a common choice that adds orthogonality constraints to the generic matrix decomposition that's on screen here. And the orthogonality constraint is what enables us to get a unique decomposition, which facilitates the interpretation. So we may see something like what's on the screen here. In a component, we see some differences, some variation between our subjects in blue that is driven by a particular microbial signature in red. Okay, so if we have repeated samples from the same subject, we can then take the time points by taxa matrix of a subject. For all our subjects K in our data, we stack them up into a three-way array, which is shown on screen. And then we can apply our tensor decomposition. So these are higher order generalizations of the PCA. And recently, approaches based on the CandyComp or Parafac, or in short, CP decomposition, have been proposed for longitudinal microbiome data in particular. And importantly, this decomposition is unique without additional constraints, so we don't need the orthogonality that we had in the matrix case. This is beneficial, of course, because biologically orthogonality between components may not be a very reasonable assumption. Now with this model, we would see something similar as before, but we see some variation in our subjects in blue due to microbial signature in red, but we also see how this pattern, how important this pattern is over time, which is shown here in green. Now, an assumption of this model is that this temporal pattern is the same between hosts, and this is not very realistic. And in particular, in some scenarios, we may be interested in quantifying or capturing shifts or delays between subjects, between each subject's pattern. Okay, to enable such structure, we propose to use the Parafac2 model. This is a more flexible tensor decomposition model than CP, where one mode is allowed to vary across levels of another mode. So as you see on screen, for each subject in the blue loading matrix, loading vector, we estimate their own temporal pattern in green. Now, this is a more complex model, so we require additional constraints. So the second line here is the constant cross-product constraint of Parafac2, which ensures uniqueness. The third line here is we additionally impose non-negativity of the subject's factor matrix, and this is purely out of convenience to fix a sign ambiguity. So with this model, we would actually then capture these subject-specific patterns. And furthermore, when visualizing real results, I will also scale the temporal loadings with the corresponding subject loadings to facilitate the interpretation of the pattern. Okay. So I will now demonstrate these approaches using data from our collaborators at COPSAC, the Copenhagen Prospective Studies on Asthma in Childhood. And in particular, we will look at the maturation of the infant gut microbiome and its relation to delivery mode over the first 6 years of life in 267 kids. Okay, so this is results already from a CP model. The slide shows one component 7, after estimating a CP model on this data. And what we see is on the left, we have a microbial signature. These are the top 15 only, truncated. And on the right, we see how this pattern differs over time between the average vaginally born and the average cesarean-born child. Overall, what we learn from this pattern is that vaginally born children's compositions are more abundant in Bacteroides, Parabacteroides, the positively loaded taxa. So Bacteroides is in purple for those who are sitting upstairs. And they have less of the negatively loaded taxa, so Clostridium, for example. And for the cesarean-born kids, it's the other way around. they have more of the negatively loaded, less of the positively loaded, relatively. Furthermore, in the scale time losing, so the right-hand side, you see how this delivery mode difference changes over time. So, of course, it's more important in the early time points, 1 week, 1 month, and then gradually diminishes as the gut microbiomes mature. Now, importantly, CP assumes that these temporal changes occur the same way in each subject, and this is a limitation that we're trying to solve. Now, if I apply ParaFact2, at a glance, we see similar results, but with a little bit more nuance. In particular, due to the added non-negativity constraint, we get a component that more describes the natural-born children and one that more describes the cesarean-born children. And for brevity, I'm only showing the former, so it's a little bit different. But as before, we see that vaginal birth results in a composition more abundant in Bacteroides and that the difference diminishes over time. Crucially, ParaFact2, however, allows us to examine these individual trajectories, which in turn show substantial variation in how the gut microbiomes mature. In particular, some kids born by a cesarean section, so the orange dashed lines, get much closer to a natural birth signature earlier than others. And this is the key kind of type of pattern that we're trying to capture. And capturing these is going to be crucial to helping elucidate what are the exposures that may underlie these differences. Okay, so a key challenge with these types of models, and this is the final thing I want to talk about, is how to select the number of components. And there exists many heuristics based on model fit, the elbow method, so extensions of two-way analysis, etc. But we use, in addition, a replicability-based approach. So here, for a particular choice of R, we estimate the model. We take our data tensor, we drop some subjects, we estimate a model, then we drop another subset, we estimate the model again. And then we take the FMS, this is a factor match score, it's a similarity score ranging from 0 to 1, 1 being a perfect match, and we compare how similar the resulting factors, the resulting patterns are between these two subsets, submodels. And then we repeat this many times with shuffling. So in prediction task, this would equate to some type of cross-validation, but unfortunately, this is not typically done with these low-rank unsupervised models. And so what we want to see is that the FMS scores are high, which would indicate that the patterns are consistent, robust, and at least representative of the study sample. Okay, so on the right, you see results of such an analysis on the CP model that I showed, and you can see that the 7-component model is still quite replicable, and there are for better explanatory power. And of course, we use other diagnostics as well, and this can be extended to ParaFact2 as well. Okay, to conclude, tensor decompositions explore longitudinal gut microbiome data. It's generic, not just gut microbiome. We propose in particular the ParaFact2 decomposition for scenarios where between subject differences in the temporal patterns are of interest. And then finally, we propose also to use replicability analysis to inform the model selection process. And with that, thank you very much for your attention. I want to thank also my collaborators.
Thank you. Thank you so much. Any questions for colleagues?
I can maybe ask one in reverse. Has anyone used any of these tools or tensor decompositions for their microbiome data before? Okay, I see a few. Great.
All right. Okay, so the speaker can also ask questions. All right. Right, but yeah.
I can ask a question. So you said cross-validation isn't typically used for setting the rank, but it is possible to do that, isn't it? By having, you sort of drop down randomly within a matrix. How does that compare to your repeatability method?
Yeah, so the question was that there is already these kind of replicability analysis done in the matrix case when you drop out elements, and indeed, and it's the same exact thing. So we're talking about the same type of approach. What I've seen typically is that at large, although it's anecdotal, with these component models, typically because of the orthogonality, we just take 2 or 3 because it's convenient, and there's no real informed component selection done. But it is indeed possible, and some people do it, but it's not very pervasive, I think.
Okay, so thank you so much for. Oh, I mean, I think I'm going to have to. Yeah, you can ask. You can ask.